In an attempt to create a balanced and self-sustaining replica of Earth's ecosystem, Biosphere 2 was a miserably and expensive failure. Numerous problems plagued the crew for almost the very beginning. Ladies and gentlemen, here at Channel 1848, we will discuss the failure of Biosphere 2, which was opened in 1991. This will include discussion on photosynthesis, cellular respiration, and a few of the colossal failures made in Biosphere 2. Other than a few domestic disputes among crew members and a large extinction event, by far the most dangerous and detrimental result of this experiment was the atmospheric imbalances within the dome. Since the sealing of the dome, Biosphere 2 experienced a steady decline in oxygen in the atmosphere. Scientists hoped that the system was merely stabilizing itself. However, 18 months in, the crew could barely function, and it was decided necessary to pump the dome with oxygen to achieve the 2-year-long goal of seclusion. This destroyed the hopes for a self-sustaining colony, as it was shown that to be unstable, but the reason why was only found years later. It turns out that the problem had less to do with oxygen and more with carbon dioxide. The soil was simply too rich and microbes were absorbing too much oxygen and pumping out a lot more carbon dioxide than expected. In plants, photosynthesis occurs in the leaves. This is where plants can get the raw materials for photosynthesis, all in one convenient location. This includes CO2, H2O, and sunlight. The process of photosynthesis is divided into two main parts, light-dependent reactions and light-independent reactions, or the Calvin cycle. The light-dependent reaction happens when solar energy is captured to make a molecule called ATP. The dark reaction, or Calvin cycle, happens when the ATP is used to make glucose. In cellular respiration, sugar is broken down to CO2 and H2O. In the process, ATP is made, which can be used for cellular work. Their overall reaction for cellular respiration. 1. Glycolysis happens in the cytoplasm. One glucose is broken down to 2 molecules of pyruvic acid, resulting in the production of 2 ATPs for every glucose. 2. Transition reaction. Pyruvic acid is shuttled to the mitochondria for further breakdown. 3. The Krebs cycle occurs in mitochondrial matrix, the liquidy part of the mitochondria. In the presence of oxygen gas, all the hydrogens are stripped off, 2 by 2, to extract electrons for making ATP, until there are no hydrogens left, and all that is left is CO2 and H2O. The Krebs cycle results in the production of only 4 ATPs, but produces a lot of NADH, which will go on to the next step. 4. The electron transport chain occurs in the folded membrane inside the chloroplast. Electrons from hydrogen are carried by NADH and passed down an electron transport chain in the production of ATP, results in the production of 32 ATP for every glucose molecule. The plant should have been able to utilize excess carbon dioxide in photosynthesis, but another chemical reaction was taking place. A large part of the biosphere was built out of concrete, which contains calcium hydroxide, and the carbon dioxide CO2 reacted with such a vast amount of concrete. This CO2 became embedded within the walls of the dome and formed H2O. This was proven beyond a doubt that the walls was tested and found to have 10 times the amount of calcium carbonate on the inside surface than the outer surface. In shorter terms, show diagram. Let us start with the producer's plants. Through this process of photosynthesis, they absorbed energy from the sun and took matter CO2 from the atmosphere and released O2. This oxygen was absorbed by the overabundance of microbes, which released CO2 back into the atmosphere through cellular respiration. The concrete within the dome absorbed the CO2, keeping some oxygen and carbon while releasing H2O back into the system. This caused less CO2 to restart the cycle starting from the plants. This caused a positive input loop leading to not enough oxygen within the dome.